Hello everyone, this is Philip Seitzer, and today I'll be talking about JContextExplorer. This is the second video in a series. If you have not seen the first video yet, which is an introduction on what JContextExplorer is and how it works, please check out that video by clicking the link appearing on the screen now. Otherwise, so I would just open your browser and go to Google, and I would type in Facciati Lab, and the first link should take you to the website of Dr. Mark Facciati at the University of California, Davis. Go ahead and go to Resources and Data Software. This will take you to a page with all of the software projects going on in the lab, and the one we're interested in right now is JContextExplorer, so go ahead and click the link, and that will start downloading a zip directory onto your computer. Okay, so I've already downloaded the file, so I'm just going to close the browser, but you would wait until it is downloaded all the way. There it is, Downloads folder. I'm just going to move it to my desktop, and then I'm going to unzip the directory. And you should see something like this. Just go ahead to the folder that says JContextExplorer package, and this is what folders look like. The program itself is in the form of a executable jar file. Jar is Java archive, and the way to execute this program is simply to double-click here. But before we go forward, I want to explain some of the other files that I included here. So I'm just going to close the program and check out the README file, which we won't open now, but you can. It gives some background to software. Go ahead and open data. So the key piece of data here are the annotated genomes. So the way that JContextExplorer works is you need to load up one or more annotated genome files, and these are in the format of GFF, or General Feature Format. So here's an example GFF file, where every line is a particular annotated feature in the genome. This is a serine threonine protein kinase that is on the reverse strands and spans these coordinates, and that is an example of a coding region. So you have one of these files for every organism that you want to include in your analysis. You also have a file called a genomic working set file. But this file is very simply a file that contains the paths to all the annotated genome files that you're interested in examining in this working set. In this case, there are 3 files, and those 3 files are the files here. So finally, the last bit of information here is this homology clusters file, which essentially what this is I went through and determined which genes are very similar in sequence to other genes, and I scanned from every gene in all 3 of these organisms to every other gene. I used BLAST, a basic local alignment and search tool, as well as NCL, or Markov clustering, to generate this file. The way to read this file is a tab limit 5 format again, the name of the organism, the contiguous segment, and then the start and stop, so the span of the genome, or the DNA, that this feature is, and then an ID number. So the gene that is located at these coordinates in this organism on this segment has ID number 1, and the gene in this organism, talking about this segment, spanning these coordinates, has ID number 548. Okay, so now let's go ahead and reopen the program one more time. And this first thing we're asked for is a genomic working set. And if we try to continue without loading these files, we are not allowed to. So let's go ahead and load up a genomic working set file. And let's go ahead, and it's going to be this one. So this is genomic working set. And then for this homology cluster that we're going to load, we don't have to, but I recommend that you do, if you can. This file. It takes some time for that to load. Then you click Submit. And now you are in the mainframe of JContextExplorer software. Okay, this completes video 2 tutorial. In the next video, we'll actually start using this. Thank you for watching.